Welcome! In this lesson, we build an intuitive picture of amino acid metabolism. We will start from structure, move to classification, then see the main reactions, and finally connect organs through the urea cycle, kidney ammonium production, and hormonal control. At the core, an alpha amino acid has an amino group, a carboxyl group, a hydrogen, and a variable side chain all attached to the same carbon, the alpha carbon. The general formula reads NH2COOH R-CH. Glycine is the simplest example. The side chain is just hydrogen, so there is no asymmetry. Proline is special because its nitrogen is tied into a ring, an imino feature. Taurine is also called an amino acid by convention, but it has a sulfonic acid at the beta position instead of a carboxyl. Except for glycine and taurine, most amino acids have a chiral alpha carbon and occur as L or D forms, with L forms dominating in mammals. There are three useful ways to classify amino acids. First, chemical families group similar structures like aliphatic, aromatic, sulfur-containing, and so on. Second, metabolic behavior distinguishes glucogenic, ketogenic, or both. Third, nutrition separates essential from non-essential, with some becoming conditionally essential. A general reaction present in many tissues is transamination. It transfers an amino group from a donor amino acid to a keto acid acceptor using an enzyme with a PLP cofactor. The mechanism is ping-pong. The donor binds, leaves its amino group on the enzyme, then the acceptor arrives and picks it up. The amino group can also be released by oxidative deamination. A classic case is glutamate dehydrogenase, reversible depending on whether ammonia or alpha-ketoglutarate is being used. Non-oxidative decarboxylation removes carbon dioxide to form bioactive amines such as histamine, tyramine, and putrescine. After removing nitrogen, the carbon skeletons converge to a few fates. They can feed the Krebs cycle, produce glucose if glucogenic, or produce ketone bodies if ketogenic. In the small intestine, nitrogen is absorbed mainly as free amino acids and as short peptides. The peptide carriers PEPT-1 and 2-MOV-D and tripeptides efficiently, and specific amino acid transporters handle neutral, imino, dibasic with cysteine, and dicarboxylic groups. Enterocytes themselves use glutamine and glutamate heavily for energy. The liver and muscle share the workload. After a meal, muscle takes up amino acids to build protein. Between meals, muscle releases alanine and glutamine, which the liver converts to glucose and urea. Branched-chain amino acids largely bypass the liver at first. Muscle transaminates them to keto acids, which the liver can use, and leucine carbon can support ketone production. Surplus nitrogen cannot be left as free ammonia. The liver converts it to urea in periportal hepatocytes through 5 steps: CPS1, OTC, ASS, ASL, and arginase. And acetylglutamate activates the entry step. Downstream, perivenous hepatocytes buffer ammonia by synthesizing glutamine. This zonation lets the body balance urea formation with ammonia handling. The kidney produces ammonium to defend acid-base balance. Glutamine is converted to glutamate, then to alpha-ketoglutarate and ammonia. Ammonia diffuses into the tubule lumen and traps protons as ammonium, which cannot come back. Hormones coordinate the flow. Insulin after meals drives amino acid entry and protein synthesis. Growth hormone helps spare protein in fasting. Glucagon and cortisol favor using amino acids for glucose and increase muscle release. In illness, cytokines amplify this catabolic push. Across the life cycle, priorities shift. In neonates, the intestine helps build arginine by expressing the middle steps of the urea pathway and using ornithine aminotransferase in reverse. In older adults, more amino acids are held by the splanchnic organs after meals, and muscle responds less to insulin and leucine, contributing to sarcopenia. To summarize, structure explains classification, transamination, deamination, and decarboxylation shape the chemistry, and organ systems coordinate nitrogen removal and carbon fuel distribution. Keep the big picture in mind. Amino groups move by transamination, carbon skeletons converge, the liver makes urea, the kidney makes ammonium, and hormones set the direction.